Welcome to possiblemedicowallah. I am going to discuss about glycolysis, which is also a universal pathway for glucose breakdown. It is also known as EMP pathway. It contains 10 steps. In 10 steps, 3 steps are not reversible and the rest 7 steps are reversible, and the enzymes are present in only cytosol. This is the pathway. This is the first step, glucose to glucose 6-phosphate. This is the one of the 3 step. This is first irreversible pathway. It is catalyzed by hexokinase, and also there is another enzyme, glucokinase, but glucokinase is present in only in liver, and also it has only power to phosphorylate glucose, but hexokinase can phosphorylate every sugar. And the inhibitor, if glucose 6-phosphate produced more, then hexokinase enzyme will be inhibited. Then come to second step. In second step, glucose 6-phosphate is converted to fructose 6-phosphate. That is the isomerization reaction induced by phosphoglucose isomerase. In this, aldose, ketose conversion occur. Means glucose is aldehyde group and fructose is ketone group. In the third step, fructose 6-phosphate converted to fructose 1,6. In this, ATP loses one PI group, that is the phosphor group, and add to fructose 1,6, and it is done by phosphofructokinase, and it is a allosteric enzyme, and it is activated by fructose 2,6 bisphosphate and AMP, and inhibited by citrate and ATP. Come to fourth step. In fourth step, fructose 1,6 bisphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone in equal concentration. It is induced by aldose enzyme, and it is the reversible reaction. This is the key intermediate step. This is the key intermediate step for preparatory phase. Then we require only one type of trioses for glucose production, that is the glyceraldehyde 3-phosphate. So dihydroxyacetone is converted to glyceraldehyde 3-phosphate by using triose phosphate isomerase. Then we got 2 glyceraldehyde 3-phosphate, means every these things are in twice. Come to sixth step. In sixth step, glyceraldehyde 3-phosphate is converted to 1,3-biphosphoglycerate by using glyceraldehyde 3-phosphodehydrogenase by using NAD+, that is the reducing agent. In this, it is converted to NADH, and NADH is the source of ATP by using ETC, means electron chain that occur in mitochondria. In NADH, our cell extract 3 ATP. as it is in twice, so 6 ATP formed. Come to next step. 1,3-biphosphoglycerate to 3-phosphoglycerate means 1 phosphorus is removed by using phosphoglycerate kinase. In this, what happened? ATP is converted to ATP. This is known as substrate level phosphorylation. Substrate level phosphorylation means ATP is the substrate and addition of pi group occur in ADP. Come to next step, in which 3-phosphoglycerate is converted to 2-phosphoglycerate by using phosphoglycerate mutates. This is a type of isomerization reaction. It is also a reversible reaction. This is a key intermediate step for the production of pyruvate. Come to next step, 2-phosphoglycerate is converted to phosphoenol pyruvate. It is in presence of enolase. Enolase enzyme is the enzyme that is inhibited by fluorides. Means fluorides can inhibit this step, and enolase requires magnesium 2 plus H cofactor for action. Then come to next step. PEP means phosphoenol pyruvate to pyruvate means removal of this phosphoenol group. It is done in presence of pyruvate kinase. This is a non-reversible reaction, and this is the important step. In this, the phosphor removed is added to ATP and converted to ATP. That is the ATP to ATP. This is the substrate level phosphorylation. Pyruvate enzyme, that is the enzyme for 10th step, that is the important for formation of pyruvate, is activated by fructose 1,6 biphosphate and AMP and inhibited by acetyl coenzyme A and ATP. Then come to ATP production. This is the 1 to this glyceraldehyde. This is the preparatory phase. This to this glycerol 3-phosphate to pyruvate is payoff phase, means it is used for ATP production. First come to ATP production. In this 2 NADH is produced because this is a triose and this phase are in 2 NADH. In this substrate level phosphorylation occur and 2 ATP is produced. In this also same process. This is the ATP produced. This is the preparation phase. In preparation phase, in this step, ATP is used for phosphorylation, and in this step also ATP is used for phosphorylation, means total 2 ATP is used. So net ATP is NADH plus 4 ATP minus 2 ATP, and each NADH is responsible for 3 ATP. 3 ATP is produced in electron transport chain that occur in mitochondria. Means total 2 into 3 is 6 ATP plus 4 ATP minus 2 ATP. Total is 8 ATP produced. But if oxygen is not present, then only 2 ATP produced. Means only this step will happen. This step will absent. So in aerobic condition 8 ATP, in anaerobic condition 2 ATP. These two occur in our body only. This occur in our heart. This occur in our muscles. Muscle like leg muscles. After exercise, lactic acid produced along with 2 ATP. Okay. Till now, what I have discussed, I will give notes on the description link. Please refer that notes, and if any doubt come, then comment. And thank you for watching. If you like the video, then subscribe also. Thank you.